Good evening, everyone. Yeah, Devdatta, you'll introduce her and you'll start the class.
Okay.
Okay.
Good evening, everyone. Today is the demo class for biotechnology. Today's class will be taken by Ms. Roshni. She is a PhD scholar at TNU Coimbatore. So I welcome Ms. Roshni to carry over the follow the class. Roshni, please share the screen.
Yes, thank you.
Please share the screen and start the class.
Is this visible?
Yeah, it's visible.
Okay. So good evening. Good evening, everyone. As she introduced me, I'm Mary Roshni from PhD Biotechnology. So since this is a demo class, we'll go for a basic concept which everyone knows, that is DNA replication. So in this demo session, I'll be dealing with the outline of DNA replication, and I'll not be dealing with all the interior topics. So first,
Roshni, put it in slideshare mode.
yeah, sure.
Yeah, please carry on.
Okay, thank you. So now, why do we need a cell to go for DNA replication? So since as all the living organisms goes by or lives by cell division for their growth and metabolic developments and so on. So for a new cell, there must be some DNA which is exactly the same copy of the one which is found in the previous or the old cell. So for that reason, we need the DNA to replicate. So there were three hypotheses. So the first was semi-conservative replication. I mean, this is not the exact order, but to say the first is semi-conservative replication. The next one could be conservative, and the next is dispersive replication. So mostly everyone will be familiar with what is all these, but to add some points, here in conservative, you'll be having a replicated newly formed DNA, which where the pairing is with between the two old strands and the two new strands, and that means the old strand is conserved and the new strand is conserved for in the further generations. And in the semi-conservative, in each and every step, the old strand and the new strand will be conserved. That is, there will be a base pairing between the old strand and the new strand. And in a dispersive, in the dispersive mode, there will not be uniform base pairing when compared with the what you say, the mother DNA or the template DNA from which the replicated ones are formed. So there will be some mismatched base pairings, or mismatched base pairings in the sense the base pairs between the old and new strand, it will not be uniform. Sometimes it will be with the old and old, or it will be sometimes with the new and new, and sometimes it will be a combination of old and new. So how to test whether what is the correct mode of replication which is found in a cell? So for that, they cultivated a bacteria, that is E. coli, in two mediums which has two types of nitrogen sources. So the first one was with N14, which is called as a light nitrogen. It had around seven protons and seven neutrons. And the next is with heavy nitrogen, that is an isotope of nitrogen of N15. So it had around 7 protons and 8 neutrons. So this experiment was done by Michelson and Stahl. So they they grow these E. coli bacteria first in N15 medium, so that all the DNA, that is after further generations, the total DNA which is in that E. coli bacteria is of N19. The reason for using this N15 in the media or N14 in the media is that all the DNA is made up of nitrogenous bases, so they need nitrogen for developing for synthesizing the nucleotide and forming a DNA strand. So they started the experiment with growing the bacteria for many generations in N15 media, and later on they shifted this N15 media grown bacteria into a media which contains N14. So then they went for the so then they started to visualize the results by using cesium chloride density gradient centrifuging. So here, the reason for using this density mediated centrifuge is that this N15 is of is of heavier nature, so that there will be easy distinguishing parameters between this N15 and N14 based DNA. So first, they started with the zero generation, that is the ones which is grown in the N15 medium, and before placing them in the N14 medium. So so in the control, that is in the normal condition, that is after growing them in the N15 medium, they had only this N15 nitrogen as, as their nucleotide base. And then when they were put in that in the medium containing N14, they had an intermediate density. So here this so since it had an intermediate density, this conservative replication was excluded, because in this in conservative uh, in conservative mode, we will be not having this intermediate density, because it will be conserved. The old strand will be conserved, and it will be having the same N15 N15 one, and the new strand will be having the same N14 one. So they excluded that conservative method. And the next type then but the next further hypothesis, like semi-conservative replication and dispersive replication, was included in further analysis, because this semi-conservative replication would also result in double strands, with one strand of N15 DNA and another strand of N14 DNA. But in the dispersive replication, it would also will result in double stranded DNA with both the strands. It be having a mixture of N15 and N14 DNA. I'm sorry. N15 and N14 DNA, but as either of this could be appeared in any of the intermediate density. So they included the semi-conservative and the dispersive mode of replications for the further analysis. And when they went for the next, that is the second generation, they found 50% in N14 and 50% in N15. So here, this was inconsistent with a dispersive type of replication, because it would have resulted in a single density, which is lower than that of the intermediate uh, when uh, when compared with the one generation of the cells. But it was still higher than the cells which was grown in N14 DNA medium, that is while comparing this is by comparing the cells which is grown only in the N14 medium, just for comparing the results. So with that, they came to know that there was no dispersive mode of replication, and that hypothesis was also rejected, and it was confirmed that replication follows semi-conservative method. So, for DNA replication, so the DNA has to unwind and it has to copy each strand, and it must create some identical copies so that the same genetic material is replicated in the further cells, and this mode of replication is semi-conservative. Then to start with a replication, you must need a replication fork. So there is this replication forks are the sites where the DNA replication starts. So this can be either unidirectional or bidirectional. So unidirectional is in the sense you'll be having the synthesis will be in only one direction, which and the origin from the origin of replication. But in bidirectional, you'll be having two replication forks which can move in opposite directions. This bidirectional replication is the one we can always see in the diagrams or pictures where you can see uh, which represents the replication. So the next is replicon. Replicon is a term is given. This replicon term is given to the DNA when this DNA is under the control of the origin of replication. So the next is what are all the proteins or enzymes which is needed in DNA replication. So you need helicase, single stranded binding proteins, primase, polymerase 3, polymerase 1, ligase or ligase, then topoisomerases. So first, so what is helicase? So this helicase is the one which unwinds the template DNA or the parental DNA. So once there is a replication force, it creates a small gap. So in that gap, this DNA helicase itself inside it, and it starts to unzip the um, two binded strands. But when you open a DNA, like when the helicase starts to unwind the DNA or unzip the DNA, you might find some places like where you can find supercoiled natures. So when this supercoiled nature is there, there will be a tension in the DNA molecule and also in the cell. So to remove this, you need an enzyme called topoisomerase. So these are topoisomerases or enzymes which can cleave the sugar or phosphate backbone of either one strand of the double helix or two strands of the double helix, which is done by topoisomerase 1 and topoisomerase 2, or it is also called as gyrase. So this allows the supercoils to unwind so that the tension which is created due to unzipping of the DNA or due to the helicase is removed. So then whenever you have, as we all know, this whatever the genome be, the size is very much big, and to complete the um, total replication in a minimum time or in few seconds, you need some 2 to 3 or multiple replication forks or bubbles to do the replication of a DNA. So for that, for example, here we have three origins of replication and three replication forks which binds to the origin of replication. And since this forms a broader space, it is also called as replication bubbles. And here the two new daughter strands form, and once these come in contact with each other, then the replication forks disappears and the two new strands uh, come to place. The next is what is the role of single strand binding proteins. So these single strand binding proteins or a group of proteins which can prevent premature annealing, and it also makes them safe from that is, it also makes them it also protects them from degrading enzymes, so that the single-stranded DNA is kept safe until the replication ends. The next is you need DNA polymerase 3 for extension of the DNA strand. So for this DNA polymerase 3 is consists of many several other proteins. So the entire protein complex is termed as the holoenzyme. So it has alpha, epsilon, theta, eta, gamma, psi, everything. And this, but the main property of this DNA polymerase 3 is that it can only add nucleotides to the free 3' end of the 3' OH end of the nucleotide chain. So you might ask where that normally there not be any free or any free 3' OH group. So where this first 3' OH group comes from? So this comes from an RNA primer, which is added by an enzyme called primase. So this primase binds to the template strand and synthesizes an RNA primer. So once this RNA primer is synthesized, and once the DNA polymerase finds some free 3' OH ends, this joins to that 3' end, and it starts to extend the nucleotide chain. And once this is formed, this primase moves out. So here the direction is 5' to 3', and the one which is that is the own strand is called as the template. Is called as the template strand. Okay, then the DNA. Okay, this I have said because that is the DNA polymerase 3 extends the RNA primers in the 5' to 3' direction. Okay, this is also called as primosome. So this primosome in E. coli has four components in it. That is DNA helicase, DNA B, that is a protein, and DNA G is also a protein, and primase enzyme. This is the primosome complex. So this primosome initiates the DNA replication or DNA synthesis at the site of replication or the site of replication fork. Then the next is this DNA polymerase 3 has the proofreading ability, because if some by mistake or something, if some other nucleotide is base paired with template nucleotide, and to remove it, it has 3' to 5' exonuclease activity. So 3' to 5' in the sense it will be moving in the 5' to 3' direction. So if by mistake one nucleotide is added behind, it can move back in the 3' to 5' direction, and it can remove that, and it can add the correct base in that position. So this mismatches is recognized by either the shape of the nucleotide or the instability um, or instability, in the sense it will not be complementary in base pairing. So uh, that can be recognized as mismatches, and this can be rectified by the proofreading ability of this DNA polymerase 3. So normally in a replication process of a double stranded DNA molecule, you will be having a continuous replication in one strand and a discontinuous replication in the another strand. So the leading strand, which has a continuous replication, is called as is the leading strand, and the other strand, which has a discontinuous replication, is called as the lagging strand, where you can see the Okazaki fragments. So why these discontinuous replication or Okazaki fragments forms in the sense? That is because both the strands, that is both the continuous replication and the discontinuous replication, moves in the direction of 5' to 3'. Okay, but as they are complementary in nature, the both strands, the first strand will be from 5' to 3', and this lower strand will be from 3' to 5'. So the direction of replication will also be in opposite side, but the 5' to 3' direction remains the same. The arrow, if you can imagine an arrow in the opposite direction, the arrow head of the arrow will be on the opposite side, but uh, when you see from the arrow side, the direction will be from 5' to 3'. So in the lagging strand, there will be some short synthesized separated fragments, which is called as the Okazaki fragments. So this as we all know, this double-stranded DNA is unzipped simultaneously while the DNA polymerase 3 starts to synthesize the new strand. So just imagine like you are unzipping in the right hand, and imagine your left hand is the direction or is the DNA polymerase 3 which acts. So your left hand moves into the right hand, and simultaneously your right hand grow or moves apart. So this 5' to 3' direction, that is the lagging strand, will move continuously and there will not be any end. But in the lagging strand, why it moves for example, if it moves one or two nucleotide in the 5' to 3' direction, you will be struck with the replication fork or with the double stranded DNA or the parental DNA, which is still double stranded. So there the RNA primer or the primase activity has to start again, so that because this DNA polymerase 3 removes from that, gets itself removed from that place, since it has acquired another base neighboring to it. So again, once the double stranded DNA starts to unwind, there is a need for a fresh OH, a 3' OH end. So for that reason, again the RNA primer comes and fits the primer there, and after that, this DNA polymerase 3 starts to move towards the 3 dash moves towards the 3' end to synthesize the new strand. For example, you can see here. So this is the leading strand. If the replication fork starts here, it moves in this 5' to 3' direction, and it moves, and that is this is the direction of the replication fork. So in the leading strand, at the 5' end, to create a 3' OH for the DNA polymerase 3 to act upon, this in this 5' end, this primase forms an RNA primer, and from that the continuous process goes. Since this moves and since the replication fork moves towards right, and this direction is also towards right, there is no stop, or there is no stop signal for the DNA polymerase 3. But in the lagging strand, this direction of the replication fork, that is the movement of the replication fork, is opposite to that of the direction of the synthesis of the strands. So, for example, if the you have a replication, for example, you can see this thing. So here, this there is a imagine here the replication fork was there in this position. So once it is moved till here, so that in this 5' end, this RNA primer is formed by the primosome complexes, and here and from here, the direction of the synthesis of new strand goes towards the 3' direction, and it will be going only by this way. It cannot go back here. So again, once this replication fork comes here, there is another need for RNA primers to get started by this DNA polymerase 3, and this becomes the reason why this lagging strand has Okazaki fragments or fragments of DNA, and as time passes, you will be having another Okazaki fragment. So this Okazaki fragment builds up, but the leading strand goes in the straight line without any break. So in lagging strand, DNA polymerase 3 cannot remove the RNA primers because it does not have the capacity to remove the RNA primers. It has only the proofreading capacity. So once this in the lagging strand, once this DNA polymerase encounters a primase, that is an RNA primer, it automatically slips off. That is it automatically is gets removed, and so that there is a gap between the Okazaki fragment and the upcoming RNA primer. Okay, you have a replication bubble like this. So this is the lagging strand, and this is the leading strand, and for this this is the lagging strand, and this is the newly made leading strand. So when you have a replication bubble like this, and there is a this is an example for bidirectional replication force, because the origin this moves in both the directions. So now here, can you see any problem? The major problem seen here is that the green portion, which is an RNA primer. So in a organism with DNA as a genetic material, no RNA can be included in the cell during replication. So to remove that, we need DNA polymerase 1. So DNA polymerase has 5' 3' RNA exonuclease activity. That is, it can remove the RNA primers in 5' 3' direction. So it can simultaneously remove the RNA primers, and instead of RNA, it can add the correct DNA base in that particular position. But it leaves the nick, that is a phosphate sugar backbone is cut at that position, which is called as a nick. So to join that, we are in need of ligase. DNA ligase or enzymes which can join two strands or two bases which has a nick in them. So in this diagram, you can see this DNA polymerase 3 is moving in this direction, but in the lagging strand, it is encountered by an RNA primer, so that this DNA polymerase 3 moves away, and this DNA polymerase 1 comes into action, where it removes the RNA primers and it adds the DNA bases, which is corresponding to the template strand, and the DNA ligase seals the gap. Okay, this is the overall of what we have seen in this DNA replication topic. This is just an outline of what is DNA replication. Okay, what is a fidelity of replication? The fidelity of replication in the sense how much it is reliable, because the faithful of replication is very much essential for life, because if there is any mistake, then it will lead to mutations. So if there is some mutation, then it might be lethal, or it might create some problem in protein formations and in similar metabolic problems. So this DNA replication machinery has a proofreading system in it, and this proofreading system is by the DNA polymerase 3, and this removes them.
Hello, Roshni.
Yeah.
Roshni, sorry to interrupt. There is some background noise because of your earphones, I guess.
I'm sorry.
Just put it, means hold it at a place, no? There is some background noise while you're talking, no?
Okay. Now am I
Just
clear?
take care of it. There is some background noise. Can you hear me?
I can hear you, but I don't hear any
Hello?
noise here. Is this okay now?
Yeah. It's clear.
Okay, thank you. So this 3 dash to 5 dash.
Yes, it's clear.
Okay, this 3 dash to 5 dash exonuclease activity of this
No,
RNA
that was, yeah.
uh,
Okay.
removes the mispaired bases, and the proofreading system is made possible. And now, what is the speed of replication? So this pol3, in the sense DNA polymerase 3 holoenzyme, can synthesis in vitro, that is inside the living cells. It can move at a speed of 730 nucleotides per second, and when it is in under lab conditions or under in vivo conditions, it is almost 1000 nucleotide per second. So you can see some differences in vitro and in vivo conditions. I'm sorry. In vitro, in the sense, in the lab conditions, it is 730 nucleotides per second, because we'll be using synthesized primers and synthesized enzymes. And if it is in the living cells, it will be 1000 nucleotides per second. That is, it will be quite faster when compared with in vitro conditions. And this enzyme is processed both under in vitro and in vivo conditions, that is this RNA polymerase 3 enzyme. Okay, now I want you all to interact with me. So here I have this is a basic question, and I want you all to answer this question. You can either unmute yourself and answer the question, or you can post the answer in the chat box. Okay, so I now I'll read the question once, and I explain what you want to do, and what the answer, how you could get the answer, and you work it yourself, and you share me the answers. So it is given that in a fruit fly, DNA replication occurs with a single replication fork. Okay, and its speed is 2600 nucleotides per minute. Now the DNA molecule, that is here you are going to that is, the question gives you the total length of the chromosome, that is 6 into 10 power 7 nucleotide pairs. Okay, this is the basic information which is given in this question. You can read the question once if you want to better understand. And from this information, what you have to answer is, if this replication is initiated at a single origin, that is, it has only one replication fork which is around the middle of the chromosome, but you have nothing to do with that middle of the chromosome. So you have to say me at what, in how many days this particular chromosome, that is the chromosome with 6 into 10 power 7 nucleotide pairs, and with a DNA replication speed of about 2600 nucleotides per minute. So with these two, you have to say me in how many days this particular chromosome will complete its replication.
Students, you can unmute yourself and answer the question.
I'm sorry for the inconvenience. I'll put it again. Yeah. Is there any reply? Or you are, if you have any problem, you can ask. If you have any problem in this, please feel free to ask. Devdata, is there any reply in the chat box?
No, not yet. Please go ahead with the answer
I'm sorry, please come again.
and explain the question.
I'm sorry,
There
please
is
come again.
answer the question. How the question is. Hello, am I audible?
Yeah.
Yes, you are audible, and there is no questions in chat.
Okay, no answers to sir?
No answers, nothing is there.
Yeah.
Students,
See, if you have any problem in this question, you can unmute or you can give it in the chat box so that I can explain.
yes, students, please. If you're not interested to speak in mic, you can put it your questions or answers in the chat. Already there are 10 participants. None of them have not giving any answer.
Yeah, are they listening?
You please explain the question once again and try answering it.
I'm sorry, I should explain once again and?
How to answer the question.
Okay, I'll say. So here you are given with the speed that is 2600 nucleotides per minute, and you have given it is given that the total length of the chromosome is 6 into 10 power 7 nucleotide pairs. So they are asking about what is the time if there is a single origin, that is, yes, someone has raised their hand.
Yeah, one student had raised their hand. Name Ayu. I think.
Yes, you have some doubts.
Yeah,
or
I
you
got
are
one answer, ma'am.
Yes,
You
sir.
can see in chat box.
Actually, I couldn't open chat box. Please.
Yeah, around 15 to 16 days.
Okay.
Answer is around 15 to 16 days, ma'am. They're given.
Okay. Can you please explain
Jnanendra Satyapati are given.
Okay. Can you please explain how you got this answer? Please unmute and explain.
Yeah, Jnanendra, can you please explain your answer?
hello, ma'am.
Yes.
Ma'am, given that nucleotide is 2600 something
Yes.
per minute,
Yeah.
in first we convert to into hour
Yeah.
into 60,
Mm.
then into convert into days into 24.
Okay.
Uh, uh, 24 after 24 after multiplication of 24, then we got 37 lakh 44,000 something.
Mm. Okay.
Again we it is given that base pair is 600 lakh.
Mm.
So that's the way. How can I
Okay.
answer?
Then after that, what did you do? You
Uh,
got that so much days, and the length of the nucleotide base pairs is given, and after that, how did you get 16 or 15 days?
I simply multiply the days with that 37 lakh 44,000.
You multiplied or you divided it?
I multiplied
Okay. You got how many days?
15 point something, 15 to 16 days.
Okay. Okay, this is the right answer. Congratulations. And if
Thank you, ma'am.
okay, and you can also try if this is it is given that it is a single origin and it is unidirectional, but and if it is a bidirectional replication, in how many days you would get the replication fulfilled for the same chromosome?
Okay, ma'am. Ma'am, another question, ma'am.
Mm.
I can understand. Ma'am, there is a question. This uh, replication is same for both prokaryotic and eukaryotic organisms. Like, कुछ difference?
Yeah, for the replication mode is also different for some prokaryote and eukaryote species, and what the polymerase enzyme which is used is also different from prokaryotes and eukaryotes. We'll be dealing detailedly in further classes. Since this is a demo class, and I said before, this is just an outline of DNA replication.
Okay, ma'am.
Okay, now answer the question. If this goes in the bidirectional replication, how many days would it take to complete the replication for the same question?
In bidirectional.
Yeah.
Yes, ma'am. I'm trying.
Others can also answer.
Ma'am, then it would be half
Yeah.
near the days.
So it will be the half, that is 8 days.
8 days.
Okay. You said that you first started with this 2600 per nucleotides per minute. So first, if you divide this 6 into 10 power 7, that is the length of the chromosome, with that given speed per minute, you will be getting this much minutes, and this will be somewhat easier in handling the data.
Okay, ma'am.
Okay, you can go by this method also.
Yes,
Okay,
ma'am.
then the next question, you have the same criteria, that is 2600 nucleotides per minute,
Yes,
and
ma'am.
you have the same length, that is 10 into 6 into 10 power 7, that is the length of the chromosome, and if this replication takes by 4 minutes, that is, it takes 4 minutes to complete. And I'm asking how many replication force would be there, or how many origin of replications would be there, so that it can complete in 4 minutes.
Okay, ma'am.
And this is a bidirectional replication, and everyone can try. And if anyone is stuck anywhere, you can unmute, or you can post it in the chat box where you're stuck, so that it will be easy to explain. Is anyone ready with the answer?
No, ma'am. Sorry, ma'am. I can't.
Okay, any other? Okay, I'll explain. So here you are given with the please mute yourself. I'm getting my voice again. It is echoing. Okay, thank you. So here it is given that you have 2600 nucleotides per minute as the speed of replication, and the total length of the chromosome is 6 into 10 power 7. So the question is, how many origin of replication would be there, so that you can complete the total replication in 4 minutes. So imagine the total, you just whenever you have some problems like this, you just imagine a genome and it is replicating, and there are so many replication forks or something like that. You imagine a diagram, and you put all the informations which is given in the problem as the points or the phrases in the diagram in your imagination, so that it will be very easy to solve problems or related to any aspects in biotechnology. Okay, so just imagine that you have a bidirectional replication and it finishes in 4 minutes. So here in that imagination, you need to find how many spots, that is how many replication origins will be there. So for that, you can first multiply this 4 minutes into that 2600 minutes, that is the speed of the replication. See, you are multiplying this given speed, that is 2600 nucleotides per minute into 4, because this it takes total replication takes just 4 minutes, and this is bidirectional one. So that is, it is multiplied by 2. So totally you'll be getting 20,800 when you multiply all these things, and you need to find this is the total time. And now you have I'm sorry, this is 10 power 7. So you have 6 into 10 power 7 nucleotide bases in the chromosome. So when you divide the total length by the total time for this bidirectional, that is 4 minutes. So when you divide these both, you'll be getting 2885. That is, the particular chromosome has 2885 origin of replications in it to complete in 4 minutes. That is, to complete the replication in 4 minutes. Do you have any doubt in this?
No, ma'am.
Okay. Do any other person have any doubt? If you have any doubt, you
No,
can.
ma'am.
Okay. Then thank you. Thank you for your patient listening. I hope you understood something. Thank you. And thank you for this opportunity.
Thank you, ma'am.
Well, good day. That's all for students. Thank you, ma'am. Thank you, and thanks for everyone for your kind cooperation for this lecture. And that's all for this for this demo class. At last, final call, students. Is there any other questions or any doubts you can ask? The ma'am is here. She can give you all the answers. Any questions regarding biotechnology? Ma'am is ready to answer for you. This is the final call. Hope there is no questions from anybody. Anyhow, ma'am, thank you, ma'am. Thank you for your support of this class.
Thank you, sir.
Yeah, Piyush Kumar, you have unmuted. You have any questions? Oh, no. Thank you. Anyhow, students, this is for all, and very soon we are going to start our regular classes of Agri Challenge Academy. Well, the classes, regular classes will be somewhat very trimmed and very moderate manner. Very good to listen. It will be completely, it will be a different era of teaching. If you have no doubts, you can leave the channel, students, or you can ask any questions regarding with this academy, everything. If you have no doubts, you can leave the channel. Thank you. And finally, students, we have given all our information regarding ACA. Just now we have uploaded our PDF with all the details. Also, we have given some details of a fee structure, what are the courses we are going to deal. You can check in the chat box. Right, I think so that's all. Thank you, ma'am. We are going to end the meeting. Thank you very much.
Thank you, sir.